Good afternoon, everybody. I am not Dr. Hirsch. Dr. Hirsch had to excuse himself because of a different commitment. My name is Sandrine van Schaik. I'm the Vice Chair for Education in the Department of Pediatrics. And I'm actually really happy that I get to introduce our speaker today, because our speaker today, Dr. Cristina Alvira, is professor in our division of pediatric critical care, as well as the Director of Physician Scientist Development for the Department of Pediatrics. And it always just really pleases me to say this, although maybe that's not politically correct, but we kind of sort of recruited her away from a different institution, not too far away. And we're so incredibly pleased to have her here. She is supported by multiple NIH R01 awards, leading a rigorous translational research program on understanding lung development. Her lab uses advanced single-cell genomics to profile molecular mechanisms in the final stages of lung growth, with the goal of developing novel therapies for infants and children with lung diseases. And today she will discuss how lung cell diversity changes after birth and how distinct cellular subpopulations respond to injury and direct repair. So please join me in welcoming Dr. Cristina Alvira.
Thanks. Thank you so much, Sandrine, for that really wonderful introduction. Okay, so for today's presentation, I'm going to talk a little bit about just lung development in general and why the immature lung, although vulnerable to injury, also is extraordinarily well poised for regeneration. We're going to talk specifically about the impaired lung and vascular development that we see in patients who develop bronchopulmonary dysplasia. My lab's interest is really on the role of the pulmonary endothelium in promoting distal lung growth. And as Sandrine mentioned, advances in how we sort of understand lung vascular biology using new methodologies like single-cell transcriptomics. So I'm not sure it's always as well appreciated as it should be that the lung is very different than many other organs like the heart or the liver, which complete their development in the first trimester of pregnancy. So the lung is continuing to develop all through pregnancy and for the first 10 years after birth. And much of this postnatal development involves this last stage of lung development called alveolarization. And this is where you have division of these primitive lung saccules into millions and millions and millions of alveoli. And this is really important, as you can appreciate, for increasing the gas exchange surface area of the lung. And this process of alveolarization occurs via what's called secondary septation. And this involves a really finely coordinated number of events among multiple different cell types within the alveolar niche to allow for directed cell migration and proliferation that allows septation of these larger saccules into millions of alveoli. And so the immaturity of the lung during all of pregnancy and after birth then heightens the susceptibility to injuries that can completely disrupt this developmental program. And I became interested in this field as a pediatric intensive care physician because a lot of the things that I would encounter taking care of babies in the PICU, like mechanical ventilation, high amounts of oxidative stress, infections, corticosteroids, are known to disrupt alveolarization. And injuries happening well before alveolarization begins, like infections in utero or intrauterine growth restriction, are also known to disrupt development of the lung. And I would say much of what we know about how alveolarization occurs has really been learned by studying the disrupted lung development that occurs in babies who develop bronchopulmonary dysplasia in the setting of preterm birth. So this disease, bronchopulmonary dysplasia, was described more than 50 years ago now. We actually had at Stanford a 50-year anniversary of bronchopulmonary dysplasia. And the first author on this New England Journal paper, Bill Northway, is actually a pediatric radiologist. And when I was an intern, actually, this was, I'm going to date myself, but this was before you had digitized images. So like when you would do radiology rounds, you would actually go down to radiology and look at physical x-rays with the radiologist. And I would do that with both Bill Northway. And actually, he was still alive when we had this 50th anniversary and was actually present at the symposium. But even back then, I think he was so insightful in this initial description of the disease, where he says it appears to be a prolongation of the healing phase of respiratory distress syndrome, combined with generalized pulmonary oxygen toxicity involving mucosal, alveolar, and vascular tissues. So what's happened over the past couple of decades, however, is that this disease has really changed from one of acute lung injury, like Dr. Northway described, to one of arrested lung development. And that's because of the really incredible advances in neonatal care. I would say most notably surfactant replacement therapy and antenatal steroids that have now allowed our neonatal colleagues to save babies born at 22 or 23 weeks gestation. So now what we see in BPD is this sort of histology, which we call alveolar simplification. And that basically means you have a lot fewer and larger air sacs. So again, that's going to overall decrease the amount of gas exchange in the lung. This disease, BPD, remains the most common complication of preterm birth. It affects 15,000 babies in the United States every year, and up to 55% of extremely premature babies. And then you can see in this graph, and you know this from taking care of these babies, it's really the smallest and the most premature babies that are most severely affected. A subset of infants will also develop pathologic pulmonary vascular disease, and this includes both pulmonary arterial hypertension and a smaller subset that develop pulmonary vein stenosis. Both of these complications increase mortality for babies with BPD, and that's particularly true for pulmonary vein stenosis, where you can see here in this single center retrospective study that was performed by Martina Sturer here at UCSF a few years ago. But it's important to note, especially for pulmonary arterial hypertension, most of the mortality actually happens early, within the first 6 to 8 months. So generally, if the babies survive past that point, you can see here from this like flat line, generally then their alveolar and vascular disease will gradually improve over time. And the reason for that is because although the immature lung is highly susceptible to injuries that disrupt development, the same capacity, the same immaturity actually increases the capacity for regeneration. And this was really first noted in rodent models where you can do a unilateral pneumonectomy, so take out the whole left lung, and that induces the right lung to actually have a process of neoalveolarization where the entire gas exchange surface area is completely recovered in 2 weeks, which is like pretty incredible if you think about it. But this capacity for regeneration is really only maximal in younger animals. So this is a study doing pneumonectomies on rats who are 3 months, 9 months, or 24 months of age. And you can see that it's only in the youngest animals where you have a complete restoration of surface area and vital capacity in that short time period. And in the older animals, it's either slower or incomplete. There's not a lot of clinical data, but there was this one study done back in the 80s where they basically followed out patients who, for whatever reason, also had to have a pneumonectomy. And they kind of separated them into age groups based on the time of the pneumonectomy. And then they did pulmonary function testing like 20 years later. And what they found was, again, it's only in the really young patients, so patients who had pneumonectomies before, when they were 5 years or younger, where they had restoration of total lung capacity almost equivalent to what you would predict for in 2 lungs. And in the oldest patients, what you see is not only do you have a much lower total lung capacity, but you have an increase in residual volume as well, which really suggests that the older patients are compensating by airspace dilation, not by generating new alveoli. And we see this tremendous capacity for infants with even really severe BPD to get better over time. And I think you can appreciate the gradual improvement of the chest X-ray here. This is a baby who obviously you can see the tracheostomy, who needed trach and mechanical ventilation for severe BPD. I think it's even more striking seeing the recovery of the lung here on CT scan images, but it takes years. And we know that from, taking care of these babies in the ICU. So you can see in this study, the average age of liberation for mechanical ventilation is 24 months, and for decannulation, 38 months. So this is why my lab really believes that if we can understand better the molecular mechanisms that promote alveolarization, that information might be leveraged into novel therapies that can sort of help increase this process, enhance it so that these babies don't need this kind of support for quite so long. So I mentioned in the beginning that secondary septation involves coordinated efforts for many cell types in the lung, and that is true. But my lab is particularly focused on the pulmonary endothelium and how the vasculature is really driving distal lung growth. And this is really in part because of what was sort of created as the vascular hypothesis of lung development, which when I started presenting this, I said recently, the recent development of the vascular hypothesis, it's actually been almost 20 years now that this vascular hypothesis was generated. And this was really coming from seminal work from Steve Abin, who's a pediatric pulmonologist at Colorado, who's really, I would say, one of the leaders, if not the leader, in the field of bronchopulmonary dysplasia, and Bernard Thébault. And what they found was that if they just looked at the lungs of babies who died from BPD, overall, the expression of angiogenic factors was down, and their sort of microvascular density was down. And then if you take animal models and all you do is directly block angiogenesis, that's sufficient to disrupt alveolarization and create the same alveolar simplification that we see in babies with BPD. And then many, many studies have shown that if you use various strategies to actually replace angiogenic factors, most frequently trying to target the vascular endothelial growth factor pathway, you can actually preserve alveolarization after injury. But it's not just the immature lung. There's been a lot of elegant work coming from Shaheen Rafi's group and others, which shows that the pulmonary endothelium is really important for driving neoalveolarization in adult models. So this is a little bit of a complex schema. So that basically what happens is, again, using a pneumonectomy model, the endothelial cells start expressing this protease called MMP14. And what it does is it basically releases all these growth factors that are normally held in the matrix that allow proliferation of epithelial and endothelial cells that allow for this coordinated sort of secondary septation neoalveolarization to occur. But what's really interesting, and that's sort of an acute injury, right? You take out the lung, one and done. This process, this physiologic process of recovery happens. But if instead you injure the lung repeatedly, and in the adult lung, often what they do is something called bleomycin. It's a chemical that induces pulmonary fibrosis. And instead, the endothelial cells, they get very dysregulated. And instead of then releasing good growth factors, they actually release things that activate fibroblasts and they drive fibrosis. So the take-home message here is that the endothelial cells are really important in sort of directing not just their own growth and growth of the vessels, but the phenotype of other cells within the lung as well. So we're fortunate because maybe the NIH wouldn't agree, but mouse models actually are really, really good models for BPD. And the reason for that is that you probably remember from that first slide, I said alveolarization begins just before birth in humans. So day of life one, term baby, already in the midst of alveolarization. Mice are actually born at the saccular stage of lung development. And they don't start alveolarization until day 5. So that allows us as scientists to actually kind of disrupt this process and really modulate it. There's also really good models of BPD in mice that are relatively simple. And the one that's most commonly used in what we use is exposing the mice to chronic hyperoxia. So if you take neonatal mice and you put them in hyperoxia, 80% for 14 days, they will develop this alveolar simplification, similar to what we see in babies with BPD. And they'll also develop pathologic pulmonary arterial muscularization. Mice are not, in general, like great models for pulmonary arterial hypertension. So this is kind of the degree of arterial muscularization you'll see in a mouse. But again, recapitulating some of the features of the human disease. And so this is why a lot of us who are interested in bronchopulmonary dysplasia and alveolarization end up using, in addition to human tissues, mouse models. So it was previously thought that alveolarization was finished at 2 years of life, and new data suggests that it probably extends to the first decade. But it is true that most of your alveoli are formed early in the first 2 to 3 years during this period that we call bulk alveolarization. And at the same time, there's just massive growth of the pulmonary microcirculation during a period of active angiogenesis. But then somewhere around 2 to 3 years in humans, and we know more certainly in rodents around postnatal day 14, as the alveolar septa start to mature and alveolarization slows, there's a period now of, instead of angiogenesis, microvascular maturation. And it's during this period where the microvasculature really switches from this phase of active growth to one of remodeling, where you have, this is a cast of the microvasculature of a postnatal day 4 rat pup. And so you have fusion of this double capillary layer, which is very characteristic of the way the capillaries form in the immature lung, then fuse to become the single capillary layer that is then within the septal wall in the adult lung. And although it's, I think, well accepted that angiogenesis is an important driver for distal lung growth, I think what's not entirely clear is whether this microvascular maturation represents kind of a break on the whole system. But what we do know is that certain injuries that we know disrupt alveolarization, for example, dexamethasone treatment to postnatal day 4 rat pups, actually induces premature fusion of this double capillary layer like weeks before it would normally occur. And so a number of years ago, my lab became very interested in trying to compare those two phases of development, one where there was active growth and one where there was sort of move to quiescence, with the idea that this would be an opportunity to kind of find ways to sort of leverage the knowledge to kind of enhance vascular growth when needed or induce quiescence when needed. So one of the biggest challenges of studying the lung is it's like really super complex. It's made up of more than 30 different cell types. And so you could imagine, for any of you all who've done science in the past, like old school ways of studying gene expression, which is you grind up the whole lung and you do a Western blot. It's probably not going to tell you what's going on between, like, a cell that maybe represents 0.1% of the cells in the lung. And so how we understand the lung has really been transformed by methodologies that allow us to interrogate the gene expression of single cells. And so this is a schema showing the most common method that we use for what we call single cell transcriptomics, where you basically digest any tissue to single cell, and then the individual cells are basically encapsulated in these little beads. Each bead has a unique barcode, and it has all the reagents for reverse transcription. So all of the transcripts that are generated then have a unique barcode for that cell. So then when you sequence, you know which transcripts come from which cells. And this method has really, I think, transformed our understanding of lots of things, but the complexity of the lung for sure. And I think one of the most stark and exciting discoveries in the past 5 years has been that the alveolar capillary network is comprised not of one type of cell, but two very, very distinct capillary endothelial cells. And this was discovered by a lot of people at the same time, but I like to give credit to Lysandra Villa-Ellis, because I feel like she was the one who really made the discovery so clearly. And what she found was in the developing mouse lung, that these two types of capillary endothelial cells were highly distinguished by the expression of this gene CAR4, which encodes carbonic anhydrase 4. So there was a population of endothelial cells with very high CAR4 expression, and then another population that didn't really express CAR4, but expressed this other protein called PLVAP. And when she did beautiful, this is like 3-dimensional imaging through the alveolus, with the alveolus circled in white, she found that these CAR4 positive capillaries are the ones that actually are covering the alveolus itself. And then the CAR4 negative capillaries are really forming the sort of capillary circles around the alveolus. And she did additional imaging. You can see these CAR4 positive endothelial cells are really large, and they have like multiple projections. CAR4 negative cells are smaller, but much more frequent. Subsequently, I would say like 3 months later, Astrid Gillich and Ross Metzger at Stanford published a subsequent paper. Here they used genetic lineage tracing to identify and label single cells. So each image, this is one single cell. So you can see here just this incredible shape of these capillary endothelial cells that cover the alveolus. And they actually are so long and so big and have so many arms, they actually stretch out and cover multiple alveoli. And they saw this and decided to name them aerocytes, which is a really cute name and very catchy. The problem is you'll hear people call them CAR4 positive cells. You'll hear them call them aerocytes. You'll hear them call them ACAPs, so aerocyte capillaries. And then LungMap, which is a group that's trying to like standardize the nomenclature for all of these different cell types, God bless them, is trying to call them CAP2s, but everyone will call them aerocytes because it was a really catchy name. So in this talk, I'm going to call them aerocytes. They called the other cells, CAR4 cell, negative cells, general capillaries. They called them general capillaries because they felt like they seemed to have a more gene expression that was more similar to other capillaries and other organs. I actually think that name is a terrible name because these are really, really cool cells. And I feel like calling them GCAPs just really takes away from that. You'll also hear them called CAR4 negative or CAP1s. The thing that's cool about these cells is they're the proliferative population. They're the progenitors. They're the repair cells. Both of the cells express really, really different genes. So you'll see in some UMAPs I'll show you for a single cell, they separate very, very clearly because they are so different, which again just highlights how their function is so distinct. And because they seem to have really different roles, aerocytes seem, based on their position, their location, the way they look, really important for gas exchange. These are the progenitors, the repair cells. Understanding what's allowing them to make the fate decisions between being one versus the other is really important, not just for BPD, but for any disease where you have disruption of the alveolar capillary membrane. So for example, for ARDS, you're going to want to balance whether you're repairing, proliferating, growing new vessels, and then terminally differentiating to become gas exchange cells. So this has been kind of the focus of our work over the past couple of years. The other thing that Lysandra showed that was really kind of interesting is that these aerocyte capillaries really require VEGF derived from the epithelium for their specifications. So what she ended up doing is she created a knockout mouse where all the alveolar epithelial cells could no longer express VEGF-A. It's this VEGF knockout. And when she did that, what she found, this is a UMAP visualization of single cell data. So you'll probably see people presenting these a lot. So on these plots, basically every dot is a single cell. And based on your transcriptome, you'll embed on the map. And so cells that have similar transcriptomes will embed closer together, and cells that have different transcriptomes will be farther apart. And then there's computer algorithms that say, okay, this is a cluster of cells that's similar enough that's probably the same kind of cell. And so you can see her cluster of CAR4 positive endothelial cells, which is here in the wild type mouse, is completely lost in the knockout mice. Again, suggesting that these cells, if they don't have epithelial derived VEGF, they're not specified. These mice also had kind of a BPD phenotype, this alveolar simplification. And that allowed the authors to conclude that the aerocyte capillaries are necessary for alveolarization. However, I think that's really hard to say because the mouse itself was also lacking VEGF throughout, which is really important for all endothelial cells. So whether or not aerocytes themselves are really important in maintaining a homeostatic alveolar niche, whether they're important for alveolarization was not really, I think, concluded from this study, but very intriguing. So I'm going to talk to you about, this is just kind of a summary of some of the work that we're doing in our lab, trying to understand the mechanisms that control capillary cell fate, and a more direct way of trying to understand these aerocyte capillaries and what they're really doing themselves for the alveolar niche. And then we have another project that's really looking at cell-specific responses of the pulmonary endothelium to injury, really focused on pulmonary venous endothelial cells, which I think are a totally understudied but super cool cell that I don't have time to talk about today, but I'm happy to talk to people about it offline or afterwards. So we, like everyone, jumped on the single cell bandwagon right before the pandemic, which was actually really, really fortuitous because we generated all this data right before. So we were actually shut down at Stanford for 9 months, totally, completely shut down, nobody going into the lab at all. But we had all this data to mine, and that was really, really cool. we set up this initial experiment because we were really interested in two transitions, like what happens to the endothelium right after birth. Because if you think about the birth transition, it's a huge, huge physiologic change. If you think about what happens, like the first breath, pulmonary vascular resistance drops, pulmonary blood flow increases something like 30-fold, cyclic stretch increases oxygen. It's a huge change for the cells. So we wanted to profile cells right before birth and right after birth. And then we also wanted to profile endothelial cells during that phase of active angiogenesis compared to the time when the cells are kind of settling down and being quiescent and helping to remodel. And this is our UMAP where we just were looking at the endothelial cells. And again, you can see each color is basically a cluster of cells that the Leiden algorithm has identified as being similar. And we were able to identify 9 different clusters of cells across these stages in development. And I think it's important to recognize if you look at this UMAP, so it's just the same data, but now it's just visualized by the developmental time point from where the cell came. And what you can see is how much the phenotype of the endothelial cells are really changing over development, because you've got some clusters comprised of only cells from a specific time point or 2 specific time points. And for us as pediatricians, we always say like kids aren't little adults, and it really is the case. So what we did here is we took our cells in yellow and we harmonized the data. So we analyzed all the data together with data from endothelial cells from tabula muris, which is the murine cell atlas. So this is adult endothelial cells from the adult mouse lung. And you can see there was like, even with adjusting for like doing batch correction, et cetera, there was really no overlap between the immature endothelial cells and the mature endothelial cells. And that was different than what we saw in the mesenchymal cells, where at later stages in development, they were actually very much like their adult cells. And then if you look at this UMAP plot again, looking by developmental time point, you just see these beautiful developmental transitions. So this large cluster in the middle is the GCAP cells, are the most frequent endothelial cell in the lung. And you can see how they embed, the embryonic cells, P1, P7, P21, kind of extending towards their adult counterparts. So we, like everyone, was super intrigued by this whole aerocyte GCAP discovery. And so the first thing that we wanted to understand was, well, when do these cells really emerge? When do these aerocytes really emerge? And when are they really specified? And this is a project that's being led by Rebecca Zhang. She was a postdoc in my lab at Stanford, and she's an assistant professional researcher here, and my superstar computational biologist, Karsten Knudsen. And if you actually go on ChatGPT or even just Google and say, like, when do aerocytes emerge? It'll say E17.5, because one person published one paper with one figure in the supplement that showed one cell that had one like transcript of CAR4, and that is like now being perpetuated in the literature till the end of time probably. We were kind of confused by that because we had this E18.5 time point and we had one capillary cluster. And I told you these cells are transcriptionally super different, so it's not hard to see them separate when they separate. So we decided to reanalyze another. This is a lot of what Karsten does. He just goes back to other people's data set and finds all these cool things that they missed, which is really awesome. But he went back to this data set that was published by Nick Negretti and Jen Sucre. It was another developmental data set that had a lot more cells and had a lot more embryonic time points. And so when Karsten just looked at the capillary endothelial cells, what he found is right before birth, none of the cells expressed TBX2, which I'll show is a transcription factor that's very uniquely expressed by the aerocyte capillaries. You'll see this little cluster of CAR4 positive cells, it's true, but they don't really separate from the rest of the cells. And when you look at another marker for aerocyte capillaries, apelin, that was actually pretty widely distributed across the cell population. And that's in contrast to, so we had a P1 time point. They had actually a P0 time point. So literally they just sacked these mice right after birth. And you can see how all of a sudden now, I think probably in response to these dramatic physiologic changes, you have this really incredible transcriptomic shift where now you do have a separation of a new population, transcriptional population of capillaries that highly express all 3 markers, TBX2, CAR4, apelin. Rebecca showed this in a different way. This is using FACS to detect CAR4 expression. And again, looking at early embryonic time points, you find that CAR4 high endothelial cells are very rare. but that right after birth, they increase and then they continue to expand over the next 2 weeks. Rebecca also wondered what hyperoxia, this model that I told you that many of us use to sort of simulate BPD, would do to the abundance of these two capillary subtypes. And what she found was that it really induces the opposite effect. So hyperoxia increases the abundance of these aerocyte capillaries, both at P14 and P7, at the expense of actually dramatically decreasing the GCAP population. And she confirmed this by reanalyzing data that was generated by Bernard Thibault's group. And again, they just had more time points and more cells, and then saw the exact same thing, that hyperoxia increases aerocyte capillaries, but decreases the GCAP general capillary population. So Rebecca wanted to understand more directly, well, what is the role of these aerocyte capillaries, particularly in alveolarization? So she wanted to create a mouse model where we could just target those cells specifically. And so what she did was she took advantage of the fact, this is a dot plot. So the darker color shows you higher expression of the gene in the different cell populations, and then the size of the dot tells you how much of the population expresses that gene. And so she used TBX2 as the gene that was going to allow her to target these cells, because you can see it's really only expressed in the capillary 2 population and not by any other endothelial cells. And it's expressed by most of the population. And so she used a mouse. And this is a really common strategy that those of us who use mouse modeling employ, where there's inducible Cre recombinase that's downstream from the TBX2 promoter. So basically, anytime TBX2 is expressed, Cre recombinase will be expressed in the setting of tamoxifen. And she bred those mice to mice that have VEGFR2 flanked by these things called LOXP sites. And so what happens is the Cre recombinase will basically delete anything between LOXP sites. So that allowed her to then only in TBX2 positive endothelial cells, which is the capillary 2 population, to delete VEGFR2, which I told you they need for survival and speciation. And so then we can just give the mice tamoxifen whenever we want to actually abrogate these cells. And so she chose to do that right after birth. So give them 3 doses of tamoxifen. And then she measured by FACS what happened to the VEGFR2 protein expression over time, just to validate that this model works. And she showed that by P7, you have to wait. You know, at P3, she only had a modest reduction, but by P7, she had a, only 26% of her endothelial cells were highly expressing VEGFR2. And she confirmed also with PCR that we were deleting. You almost never delete the whole gene in these strategies, just a really critical exon, that she deleted exon 3, which was the critical exon that's flanked in the KDR gene, which is the gene that encodes VEGFR2. And then we didn't have any compensatory increases in FLT1, because FLT1 encodes the VEGF receptor 1. So we didn't want something to like compensate for this. Oh, my clicker died. It was working so well. Okay. So she was able to show in these mice, and this is mice just, the wild type mice are here, VEGFR2 wild type, VEGFR2 delta cap2 are the mice that have lost VEGFR2 in the aerocyte capillaries. And you can see that even at P6, the early time point, we're already starting to get airspace dilation. But even if we find alveoli that look like they've already formed and looked at the structure of the vasculature, so CD31 is an endothelial stain, you can see that the vasculature is starting to get almost like this moth-eaten appearance, in contrast to the wild-type mice where you have almost complete coverage of the alveolus. The other thing that was really interesting is we saw this really tremendous increase in alpha smooth muscle actin surrounding like the alveolar rings. And that's a sort of higher image picture to see that. Rebecca also wanted to kind of better phenotype the ACAP and GCAP abundance in this mouse model. So we performed single cell transcriptomics on these mice. We ended up profiling all the different lineages in the lung, but then we just took the capillaries and embedded them alone. And when we did that from this data set, it was surprising because what we saw was actually now a third cluster, that embedded between the A caps and the G caps that we called cap intermediates, because they expressed not really unique markers, but they co-expressed markers for both of the different subtypes. And when we looked at this data, looking at each sort of experimental group individually, so each genotype and each experimental condition individually, you can see that some clusters are more represented at different in different experimental groups. So for example, if you look at just the wild type mice, which are the red ones, right? The majority of your endothelial cells are these CAP1 GCAP cells, almost 50% total ECs, a lot of aerocytes, and very, very few cap intermediates, less than 4%. And this is why they've probably been missed when we were looking previously, because it's a really rare population at P7, which was the time point we tended to focus on, especially in normoxia. In hyperoxia, what happens in the wild type mice is that you get this, gradual starting to get this increase in CAP2s or aerocyte capillaries, which I showed you before. They expand in hyperoxia, and you get a threefold increase in these cap intermediates. The knockout mice, we completely blocked CAP2 terminal differentiation. So they had almost no endothelial cells that were transcriptionally considered by the computer to be aerocyte capillaries. And instead, what happens is they have all these capillaries get stuck in the intermediate state. So in the normoxic mice, that's about 15% of their capillaries are these cap intermediates, and more, almost 30% in hyperoxia. And this was something that was a relatively new finding and very intriguing. The loss of either terminally differentiated aerocyte capillaries or the accumulation of intermediates, we don't know yet, was associated with even more profound structural changes when we took the mice out to P14. So you can say here in normoxia, you're starting to see much more apparent airspace enlargement in the knockout mice. And then in the mice exposed to hyperoxia, both groups have really profound airspace enlargement, as you can appreciate. But again, here on these higher magnification images where we're staining the endothelial cells with CD31, the endothelial specific marker, you can see in the wild type mice in normoxia, they still have pretty continuous coverage of endothelial cells along all of these dilated airspaces. But that's not what we saw in the knockout mice. What we're seeing in the knockout mice is actually we've lost a lot of capillary coverage. And instead, what we're seeing is an increase in alpha SMA expression in the distal lung, which is very abnormal. So this suggested that alterations in capillary fate wasn't just affecting the endothelial cells, but actually was inducing what we call like non-cell autonomous effects in other populations in the cell. And because they were ACTA positive, it made us think that this was probably the mesenchymal cell population. So what we did next was we did more sequencing. Once you start sequencing, you can't stop sequencing. It's like an addiction. But so we sequenced now 44,000 mesenchymal cells. And you can see, again, just like highlighting the heterogeneity of the lung, right? So all these, we had basically identified all the different mesenchymal subtypes. And again, found in our knockout mice, there was just stark alterations and just abundance. We're still going through like the phenotypic transcriptomic changes, but just in terms of abundance, we had an increase in our alveolar fibroblasts. If you look at the blue bar, that's our knockouts in normoxia. We lose our myofibroblasts. Myofibroblasts are really important for alveolarization and secondary septation. And we lose our pericytes. And we've just started mining this data transcriptomically, but the alveolar fibroblasts definitely, even at P7, this is a P7 time point, so still pretty early in the process, start upregulating pretty dramatically ACTA2. And they also start upregulating a lot of other genes that are markers for basically fibroblast activation. So again, suggesting that alterations in capillary cell fate are potentially driving fibrosis in this model. We were really kind of confused when we got this first data from the knockout mice, seeing this intermediate cluster as like, why didn't we see this before? We've been doing this for years. And we thought, well, maybe it's because it's such a rare population. And with everything, like with clinical studies, with single-cell transcriptomics, it comes down to power. And if you're looking for a relatively rare population, the only way you're going to see it is if you profile a lot of cells, number one, and also if you sequence really deep. You have to do deep enough sequencing to detect a certain number of unique genes per cell in order to really see differences. And so we redesigned an experiment at an earlier time point because we thought, well, maybe these are the progenitors and they're just present a lot earlier, like before alveolarization begins. So we chose the P3 time point in mice, so that's saccular stage of development. We profiled over 11,000 endothelial cells, and we did like super deep sequencing. So we ended up detecting more than 4,000 genes per endothelial cell. And when we did that, then this cap intermediate cluster was very obvious, and this here in the pink. The other really cool thing, I think, is that, so in single cell transcriptomics, if you have proliferating cells, which you will in the immature lung, right, because the cell's growing, the transcriptional program for proliferation is so strong that they'll just cluster with each other. Like all the proliferating cells will cluster with each other. So what we started doing is we do that initially, so then we know which ones are the proliferating cells. Then we regress, we remove all the cell cycle genes, take all of them out. And then that allows our cells to embed with their own cell type and identify where the proliferating cells would be. And when we do that, the proliferating cells are navy. You can see that they're in the CAP1, GCAP cluster, which we know that's a proliferative population, but a lot of them are in this intermediate state as well. And this is just the dot plot again, showing you the co-expression of markers from these two different subtypes and intermediates. So once we had this data, we did another P7 data set. Now this time was normoxia and hyperoxia together, using the exact same strategy. And again, we still had this large cluster of cap intermediates. And at this time point, almost all the proliferating cells are actually in the intermediate state. And I think this abundance plot shows why we missed them before. So at P3, they're actually pretty abundant. 15% of the cells are in the cap intermediate state. But again, about 3% by the time you get to P7, unless the mice are exposed to hyperoxia, and that pushes this cap intermediate population up. You always have to validate what you find computationally. So we wanted to really prove the existence of these cells. So we used a method the trade name is RNA-scope, but it's basically fluorescent in situ hybridization, where we're detecting the transcripts for these genes in these colors. And so this is a GCAP marker, another GCAP marker, and then a ACAP marker. So we were looking for cells that express all three colors. You can see this is a beautiful example here of a cap intermediate expressing all three marker genes and distinguished from the smaller GCAP that was only expressing PLVA, P, and KIT. And then in the hyperoxia exposed mice, you can see kind of like these like clusters of these two cells all interspersed with each other. That's great. That's what's going on in the mouse. Is this actually true in the humans? Like, this is a paradigm that's true for human biology. So we're always trying to save money. So we're always trying to like reanalyze people's data that's already been generated, so we don't have to do everything ourselves. So this is data that was generated by LungMap, and they took 2 human donors that died on day of life 1. One was preterm and one was term. Most of the time, these babies are babies who died of anencephaly. And this is the initial published UMAP. They weren't really focused on endothelial cells, so they didn't even subtype them. They just had this one cluster of endothelial cells there. Let's see. So Karsten went ahead and re-embedded just the endothelial cells, and again found with ligand clustering that now you have this green cluster that's embedding between the ACAPs and the GCAPs that co-expresses markers of both. So capillary intermediates are present in the adult lung as well. What was really kind of interesting, I think, is that, again, 2 samples. When you look at like the donor-specific representation, vast majority of the intermediates were coming actually from the preterm donor. So these intermediates made up about 22% of the endothelial cells in that preterm donor. We got tissue from a separate preterm donor in the Brindle lung repository, and was able to show by in situ hybridization, this time using the endothelial specific marker PCAM with the ACAP marker TBX2 and the GCAP marker apelin receptor, to find these triple positive cells. And there was kind of 2 populations. There was one bigger population that had kind of lower expression of all 3 markers, and then these really small cells that had really, really high expression And these kind of areas of condensed nuclei that made us at first think that they were apoptotic cells, but because they have such high gene expression also, we actually think that they're probably actively dividing cells. So we then wondered whether these alterations in capillary fate might be occurring in babies who die from BPD. So this is now, again, publicly accessible, it hasn't been published, but it's publicly accessible data from LungMap. LungMap is this big consortium that was established probably 7 years ago by NIH-LBI, and they to really try and use these kinds of strategies to understand all sorts of pediatric lung disease. So it's been actually really a great resource for the community. And what they did is they had 4 groups. They had patients who died of active BPD. So that was typically babies who had severe BPD and died from respiratory failure. Most of the times they were babies who never got off the vent. They had babies who died from what they called established BPD, which when you don't have as an investigator in Brindle, I guess that we're more like collaborators, didn't really have like access to in-depth clinical data, you just have a little bit of clinical metadata. So the established BPD babies tended to be babies that seemed to were still being chronically ventilated, but died from some other cause. So for example, like their tracheostomy got dislodged, so they would be in the established BPD because they weren't dying from respiratory failure per se. Then they had babies who were healed BPD, who were no longer on respiratory support. And then they had age match controls for each of the groups, because you can imagine that the ages of all 3 of these groups would have been different, because the healed BPD group is going to be a little bit older. So what we found was, first of all, that there was a significant increase in the number of CAP intermediates in babies dying from active BPD. But actually, I think what was more interesting is you can see there's like patients who have the high CAP intermediates and patients who don't, patients who have more like the same level as controls. And I think all of those of us who take care of patients with BPD recognize that it's not like every patient with BPD is the same. There's these very clear endotypes where patients have variable degrees of airway, parenchymal, vascular involvement. So we thought, well, is this almost like a molecular readout of a specific endotype of BPD baby who has like more vascular disease? So we were able, so first what we did is we said, okay, we're going to ignore their original categorizations, and we're just going to actually reanalyze the data, putting them into groups where they're either high CAP intermediates or sort of normal CAP intermediates, and analyze them that way. And when we did that, and I looked through, and it's not a robust data set. I think that's one of the problems with the human sequencing from human cadaveric tissue, especially this was done many years ago. The sequencing depth was really shallow. So there's some like populations that would have been interesting to look at, but they just didn't have the cell number or the sequencing depth. But it was clear from reanalyzing that data that the pulmonary arterial endothelial cells in the patients who have high CAP ints were very different. And many of the genes that were highly upregulated were genes. And if you just basically what I did is I like took the whole like list of the top 30. I don't even use Metascape anymore. I put it straight into ChatGPT, and I'm like, tell me, what does this suggest to you? And it was literally, it spelled like basically said, this looks like a gene signature that you would see in pulmonary arterial hypertension. And I was like, well, that's really cool. And it's because there's markers like the CXCL12, which is normally important for arterial identity, but is also a big driver of neural cell recruitment and intramural hyperplasia and PAH. You have notch downstream effectors like DLL4 and HES1, which we know prune, like stop arterial growth. So that's going to prune the microvasculature. You've got high expression of ACE, a vasoconstrictor, and then you have high expression of elastin fibrin assembly protein. So that's going to be driving sort of this muscularization and abnormal matrix formation. So we asked LungMap if we could have some lung tissue from some of these donors, and we got 2 patients that had high CAP intermediates and 1 patient that had the low CAP intermediates and 1 control. And we stained for alpha smooth muscle actin, which will stain vascular smooth muscle cells and a lot of other matrix producing cells. And you can see compared to the control, we have these like beautiful thin walled little arterioles throughout the distal lung and normal air spaces that in the 2 patients that we were able to obtain with high CAP intermediates, pretty dramatic. Actually, this patient has really, really dramatic pulmonary arterial vasculopathy. And it's really interesting because the patient who has low cap intermediates, it's not like this patient just didn't have severe disease. If anything, the alveolarization in this patient, like it was actually striking. It was so much black. Like there's like, it's kind of hard to imagine like this patient, how is this patient even exchanging gas? There's like no alveoli. And vessels they're not normal. They're definitely thicker than the controls, but nowhere near sort of what's going on here. And so then we thought, well, this is really interesting, but is this like, what's the chicken and what's the egg? Like, is it the, alterations in the capillary fate that's driving it? Is it the other way around? So we went back to our mouse model and we said, well, is there any evidence that our mice are starting to get arteriopathy? And these are sort of early studies. This is only P14. So in hyperoxia, normally you get no muscularization until P28, P42. But in Rebecca's knockout mice, she's already getting thickening of especially the small arterioles in the knockout mice, which suggests to us that there probably is something about alterations at the capillary level that's all causing sort of pathology more proximal. So this is, I would say, one of the sort of new focuses of our work. Okay, so to summarize, I think take-home points. The lung continues to develop throughout pregnancy and for many years after birth, which leaves it really susceptible to injuries that disrupt its development. The pulmonary endothelium plays a really important birth, both in development and repair, through its ability to secrete these, what we call angiocrine signals that regulate not just its lineage, but all the other lineages in the lung. I think that this kind of discovery that there are two distinct capillary endothelial cells is so exciting, but also surprising. It's taken us so long to figure this out because it kind of models the paradigm in the alveolar epithelial cells. We've known for so many years there's the type 2 and the type 1 cells. In that paradigm, the type 2 cells, right, are the progenitors that make new type 1s. So it's kind of unfortunate we named these cap 1 and cap. We should flip-flop the names, adding to the confusion. But I think they're so functionally different and they're so molecularly different that it's going to be really important for all of us to take care of critically ill patients, to think about what are the mechanisms and what are the therapies we could give to actually modulate some of these things. There's a subset of general capillaries that seem to enter this highly proliferative intermediate state, and Rebecca's working on trying to figure out like what are the initial signals to do that, and then subsequently differentiate into erythrocytes via VEGFR2. It doesn't seem like VEGFR2 is required for them to be in the intermediate state because if anything, our knockout mice have more intermediates. And we do think that loss of erythrocytes directly alters the alveolar niche and may induce pulmonary arterial remodeling. And I think this just really highlights these methodologies like single cell transcriptomics and how they're able to really, I think, reveal these complex cell populations and highlight that there are specialized populations that are really performing really important reparative functions. And this is probably true in all organs, not just the lung. So thank you for your attention. I'm super happy to be here. I'm really, really thankful that Jeff and Dr. Hirsch and Kevin Shannon, it was like the longest recruitment ever. Actually, I reached out to Jeff in 2014 telling him I wanted to come to UCSF. So it took like 10 years to put all the pieces together. I'm really happy to be here. That's my little small and mighty lab. We've been waiting to like fully expand, just trying to let the dust settle from everything that's going on in the NIH right now. We've been really, really fortunate to be well funded up until this point. We have so many awesome collaborators, and we got into single cell transcriptomics. I knew nothing about this. It was like so naive for me to even embark on this. But we partnered with this really tremendous computational biologist and physicist, Fabio Zamini, who was finishing up his postdoc in the Quake lab when I was at Stanford and just bored. Because he had like 3 months before he was taking his job in Australia and he didn't have anything to do. And so he started analyzing our data and he was like, this is really cool data. And so we still have single cell meetings every week with Fabio's group in Australia, and he's been like a tremendous collaborator. So I'm happy to answer any questions about this or about anything else. For the trainees in the audience, I think it's important to know I never held a pipette in my life until I was a fellow. I did zero bench lab work, zero. I never even worked in a lab in the summers for my resume. I did nothing. So I started in science as a fellow. and just got hooked. And so if you're interested in discovery, even if you thought it wasn't something that you wanted to do, please come and talk to me and I'd be happy to tell you all about it.
Thank you very much. That was fantastic. I don't know if there are questions. There's some questions on Zoom, but we'll start with the room.
Yes, April.
Wonderful talk. Thank you for very beautiful pictures and also sort of taking us on a journey through things I did not know that as someone who treats this entity.
Isn't it so cool that nobody knows this?
Yeah, it's very cool. And at the risk of sounding like a complete noob and really knowing nothing about bench research at all, I'm curious hearing some of the challenges with mouse models versus available human tissue.
Yeah.
Is there a role at all in any of this work for like organoids? Like, could that?
Yeah, people have been trying to make alveolar organoids for a long time. And right before I was leaving Stanford, I had a little extra money to spend I couldn't bring with me. So I started going to like random meetings that I wouldn't necessarily go to. And I was like, I need to learn more about the epithelial cells, because I had been totally ignoring epithelial cells. And then I was like, well, they seem like they're probably really important in this process. So I started going to these like epithelial lung stem cell meetings, and there was a lot of organoid work presented there. The problem with the organoids that they're currently using is they're almost exclusively epithelial cells and stromal cells. They cannot figure out how to incorporate endothelial cells, but they present it like alveolar spheres. And for the vascular biologists in the room, we're like, that you're missing like one half of the whole thing. Like, so I think we'll get there. And obviously there's a huge NIH push to move us towards like alternative models. From my standpoint, what we've decided is we're just going to lean heavy into the human and working with, so the problem with the current human data that LungMap has generated is, and this isn't a criticism, they're trying to create a resource for everybody. So they're frequently taking the lung and just taking all the lung cells for this disease or for this time point, et cetera. But when you do that, so for example, in that BPD data set, only 5% of the cells were endothelial cell of the whole data set. Okay, so then if only 5% are endothelial cells, if I'm looking at cap intermediates or venous endothelial cells, they're just not represented. So we've been working with Tom Arnold's group because they're really interested in brain vasculature, and they had been optimizing ways in which from the frozen human tissue, which you have to do then not single cell but single nuclei isolation, you can use cell-specific transcription factors to FACS sort your nuclei of interest. And so we started doing that. And so we were able, with this transcription factor ERG, which is specific for endos, we can now isolate just the endos from the frozen lung tissue. And so this R1 that I'm trying to write, that's due technically today, but it's not going to be done till Monday, is on the pulmonary venous endothelial cells, because I'm super interested in pulmonary vein stenosis. And I think this way we'll be able to generate a really robust endothelial data set from disease. And so that's the strategy we're taking, because I've seen people work in organoids. I'm just going to wait for someone to do it, and then I'll collaborate with them, because I just don't have the time. It costs a lot of money too, I think, to do them. So, but it's a really good question.
I don't know that we're going to get through all these questions, but I'm going to start with one from Dr. James Padbury. In the tamaxis knockouts, how was their somatic growth, lung function, and any neuroculture testing?
So it's a great question, Jim. Their somatic health is good. We haven't looked at their neurofunction, or we haven't done pulmonary function testing. The mouse model to really do those studies will have to be refined, because what ends up happening, and this makes sense now that we know that there's intermediates, we only give them tamoxifen from P0 to P2. So that deletes VEGFR2 from whatever intermediates and CAP2s happen to be present at that time. But then the tamoxifen wears off, and then the capillary ones send new cells into the intermediate space that then can become CAP2s. So they can recover partially themselves over time. So now we have an experiment plan where we're doing prolonged tamoxifen to try and really knock them out and keep them out. And then I think we can do that sort of longer term phenotyping.
We are at time. So Dr. Kleiman and Dr. Abakian, if you're still on, I'm going to actually suggest that you connect directly with Dr. Alvira, because I have to go back to the teacup.
Yeah.
Take care of patients with BPD.
Very good.
Thank you so much.